genes, or there are context-dependent essential genes which are required for some specific cells, and there are non-essential genes. They are obviously essential, but they are non-essential for cell viability. So after the concept of gene essentiality and context dependency, I would like to briefly talk about how a basic CRISPR-Cas9 works. So in the CRISPR-Cas9 technology, there are two components. One is guide RNA, and other is CRISPR endonuclease. So, how it works, they need to be create a complex, and in this complex, guide RNA recognize the target sequence, and Cas9 is endonuclease, and it cuts the DNA. So when this spacer sequence on the guide RNA recognize the target sequence and anneal with it, then Cas9 create the double-strand breaks on the DNA. And these breaks create the stress, and this stress activate the double-strand break repair pathways. And because the primary repair pathway is non-homologous end joining, and it creates huge indels and frame shift across the double-strand break site, it generally results in, most of the time, results in loss of function of the gene. So why is so important, and this technology won the Nobel Prize last year? Because Emmanuel Charpentier and Jennifer Doudna indicated that we can actually program this guide RNA, so we can actually specify the target sequence as our wish. So they actually developed the precise genome editing technology. So because we know that we can manipulate this guide RNA, so we can synthetically synthesize guide RNA that can target like the interested gene sequence. So researchers think that, okay, we can create lots of guide RNAs, and if we can work with them in parallel. So they create CRISPR library, which is composed of so many different guide RNAs inside it, which are target the different DNA sequences. By using the lentiviruses as delivering tool, they are infecting the cells with this guide RNA. So at the end, they are looking at the alive and dead cells and their genomic components, and then they decide that which of the guide RNAs are inside the alive cell. So which brings us the cell type specific depleted or enriched guide RNA inside them. So it could be a little bit complicated because I think I couldn't explain it really good. So in here, because of that, I draw some examples. For example, in here, the gray ones are dead cells. Yellow guide RNA targeted sequences, genes, when we knock out them, it doesn't affect the viability of these cells. However, if we look at the red targeted genes, if we knock out them, the cells are dying. So we can say that yellow guide RNA targeted genes is not important for essential, but red guide RNA targeted genes is absolutely essential. However, if we look at the green targeted, green guide RNA targeted genes, it depends on which cells we are infecting. So these results give us how essential these genes for this specific type of cells. So we are lucky because two institutes, Sanger Institute and Broad Institute, one from UK and the other from States, they do huge amount of knockout screens. So basically, they knock out each of the more than 19,000 genes one by one in more than 300 cancer cell lines. So we have essentiality scores for each genes in each cell lines. We will use these data, and I will come there. So what was our aim? Because we have data to essentiality of the genes inside different type of cancer cells, we thought that context dependency, if we use the context dependency, we can have a really good cancer therapeutics. Because if we assume that these genes under the specific context is important and affect the viability of the cell, we can basically create a drug And if we use this drug, it wouldn't harm the healthy cells, but it would kill the tumor cells. So it is really important to know which genes are the context-dependent essential in cancer cells. But we think that it is important to which are the context-dependent, but also where and how they are essential is also really important to actually diagnose and also create the therapeutics in the future. So we aim to develop a tool which can both identify the context-dependent essential genes and their underlying contexts. So which data we used? So as I said before, we use the CRISPR screens from Sangram Broad Institutes. And also these institutes has information for these cell lines and their mutational background, their expression levels, their CMV levels, fusions, and also Sangram Institutes have the drug response of these cell lines. So we collect all of these information to extract the context information. And for other data set, we use Bagel. Bagel is the gold standard essential and non-essential gene sets. They are giving, for example, the ribosomal proteins as essential, but other genes are not. This is gold standard essential genes. We use it for training our model. And then there are ADAM and human pluripotent stem cell core genes. We use them for comparison or prediction. And the other ones are to annotate the genes if they are transcription factor or surface protein, if they are kinases, they are for annotation, as I will show later. So because we have two main aims, we divide our methodology into two. In the first one, I will talk about identification of the core and context-dependent essential genes, and the second one is the contexts. So the first one, identification of the core and context essential genes. I'm coming here. So before the methodology, I would like to show a little bit of the challenges before we started this to solve this problem. So in the middle of the presentation, you can see the structure of the essentiality table. So in here, for both studies, they give in each row genes and each column cancer cell lines. And in each entity shows that the essentiality, how essential these genes in these corresponding cell lines. So we thought that if we go into, if we look into cell line by cell line across genes, we thought that there should be two patterns. The essentiality values should be high or should be low. Yeah, some of them are essential, some of them are not. In here, our hypothesis is that context-dependent essential genes, if they are in their context, they should act like core essential genes. Their values should be as similar as core ones because they are in their context. So our advantages, we can separate the non-essential to essential, but our disadvantage is we cannot differentiate core to context because we are not knowing the gene's behavior across all cell lines. This brings us, how about if we look at gene by gene, but across cell lines? So we thought that there will be three different patterns. So gene value across cell line could be all high, which brings us the core essentials, or low, all low, so it brings us the non-essentials. Or there are fluctuating values. This brings us the context-dependent essential genes. Advantages, we can differentiate all of them. But the disadvantages is that to identify context-essential genes, there should be lots of cell lines having these contexts. But this means that we can lose the rare context-essential genes. So, but we don't want it. So we think that we need to combine these two approaches in our methodology to identify all of them. So what did we do? We take Bagel core and non-essential genes and their essentiality values in our both Sangram Broad data sets. We did this methodology for two of them separately. So what we did, we trained the logistic regression model and we did our benchmarking and we found that our prediction is pretty much good. And then we applied this model onto essentiality values of non-Bagel genes, the remaining genes. So instead of taking the logistic regression binary score as success or fail, if it is essential or not, what we take is we take the probability of being essential the LR model gives us. So how probable this gene is essential in this cell line? We have these values. And in here, we use across genes because we look each cell line and we check if this cell is essential or not. I mean, how probable is this essential in this cell line or not? Then we continue with this probability table and we looked at the probability distribution of each genes across the cell lines, which brings us the across cell lines behavior of the genes. So when we take this distribution of given genes probability of being essential, we discretize it and we create the arrays of the probability distribution. We just take these continuous values into discrete values to run k-means clustering. In this k-mean clustering, what we are doing is we are basically clustering the distribution behavior of the probability of being essential. So when we look at cluster 1, for example, in the cluster 1, the probabilities are peak at 1, which means that all of these genes in this cluster are essential across all cell lines because their distribution are peak at 1. But when we look at cluster 4, there is a peak at 0, so which means that these genes are non-essentials. When we look at cluster 2 and 3, we see the context-dependent essential genes because the probabilities are fluctuating. But what is interested in here, in cluster 3, we catch very small amount of cell lines having the cluster 3 genes as their context-dependent essential genes. So it is quite good for us because we catch them, but we didn't actually miss them. Okay. And this is, in here, we use the across cell lines approach. So in this way, we merge two approaches together. So what did we get? We discovered novel core genes as well as the known core genes. And no, we predict the novel core genes and also the known genes. And second one is we label the context-dependent essential genes in our gene set. So our results. So as you can see in this Venn diagram, these are the numbers of genes that we labeled, that we predicted from our model from both studies. We put ADAM in here. ADAM is Sanger database prediction also. And as you can see here, we predicted the ADAM genes, but only 20 of them only predicted by Sanger. When we look at these 20 genes, we see discrepancies across their essentiality values for both of the studies. So because of that, broad dataset, broad model, couldn't label these genes as core. So because we saw only broad and only Sanger, because of these 20 genes, and also we saw that broad and Sanger clustering score, in here, this is the SLED score, is low compared to intersected genes. We decided to continue with core labeling as if the genes are predicted with both projects. So in here, I say SLED score. Maybe there are people who don't know the SLED score. SLED score represents how well the element in the clusters are in there. So if SLED scores is high, which means that the elements in there, the gene is in the cluster, is in their right cluster. So it represents this. So when we look at our core labeled genes, when we only take the intersection, we just look at if our predicted core genes are where, and we saw that half of them are novel, other half of them could therapeutically tractable genes in the essential cellular processes or stem cell core genes. But we observed that these half of them is novel. They did not predict it before. We looked at protein complexes, in which protein complexes they are in. We found that they are generally in the already known core essential processes. And as well as we found I didn't put here actually, but we found one of the COP9 singlasome complex as a essential complex too. So our other hypothesis is that we think that if these are contact accord genes, they should be high in normal cells too, because they are housekeeping. They are important cells for cells viability. So what we did, we collect the normal tissues basal expression levels from GTEx, and we compare these essential genes expression with the genes that we didn't label as core. And we observed from the pairwise comparison expression levels of both gold standard genes and our predictions. They are significantly different than the non-core genes. So this is from this part is core essential genes. But now I will go into the identification of the underlying context. When I say context, we have tissue and cancer types and mutation and expression and drug response data. So we use these data to identify it, but there could be like different MSI status or copy number, as I said. There are lots of contexts could be, but we use these contexts. So our methodology, how we what was our approach? So we take the essentiality values again, and also structure of the context data. When I say structure, for example, mutation data is a discrete data because there's no continuity in there. It should be mutated or non-mutated. For example, expression data should be continuous because we have a continuous number of expression levels. So according to the structure of the data, our package is choosing the proper statistical tests. It could be correlation or Wilkinson or Mann-Whitney, or for example, the comparison of several groups, we are using Kruskal values. These are all non-parametric tests because most of the time the data is non-continuous, so we use the non-parametric version of the tests. So we collect the significant association between gene itself or gene A to gene B, or gene to tissue type or cancer type. So we collect these significant information and we apply the test iteratively for all of the combinations. So this is an example results, which you can directly navigate through our website, CEN-tools.com. So in here, for example, what we did is essentiality score of BRAF and how differ these essentiality scores across different tissues. For example, in here, skin tissue, these are melanoma cell lines. We see that BRAF is essential, like other pan-cancer. They are really essential, significantly essential. You can observe this. Or even here, for example, you can search through the expression levels of, for example, SOX10, but we know that SOX10 is only expressed in skin melanoma tissues again. We see that their expression is high. Also, its essentiality is high too. So, for example, you can search through the mutation context. For example, what I'm saying for mutation, if a gene is mutated, if the gene is mutated, if this mutation affects the essentiality of itself or other gene, you can search through co-essentiality in some specific cell to pan-cancer. Or these are just an example I will explain later because of that I'm going quite fast. For example, you can search through if essentiality values is correlated with drug response or drug resistant of the cell lines in specific tissues or cell, cancer types again. And also you can search through essentiality to expression correlation of the genes. It could be different or same gene inside the tissue or pan-cancer. But these are all one-to-one association, and it is really hard to link the, how I can say, create the big picture. So what we think that we can collect all of these significant association and we can use a network approach and we can link the associated genes to each other and also genes to tissues. So we did it and we called them context essential networks, context dependent essential networks. Then we are just saying them as CENs. So these are example of CENs. For example, in here, while using Sanger study and using the esophagus tissue, these are the genes that are essentially significantly essential in esophagus cancer cell lines. For example, in here, this is the, I just put here how you can use the website too. So in here, the project is broad and the tissue is select and tissue is skin, and we are using the context as mutation. So what we can see in here, in the skin tissue, in melanoma cell lines, there are two main driver mutation, which are BRAF and NRAS. And when they are mutated, these shows the genes that are essential when, for example, BRAF is mutated. This shows that when NRAS is mutated, these genes are essential. And we know that in melanoma, that different mutations, when there are existence of different mutations, we know that signaling inside the cell is different. So it explains that why there are different essential genes when there are different mutations. So these are examples, but now I'm coming, okay, we created it, but how what is the importance of this finding? So I will only show two examples. One is the CEN-tools to pickles result comparison, and the other one is what about if we merge the cells with the known protein-protein interactions, if we give us any signaling-based results. So the first one is the comparison. The pickles is the co-essentiality networks from Hard Lab. This is the lab the bagel genes are produced. So they show that all of these genes are co-essential with BRAF, but there's a miscalculation, I said here, because in melanoma, the main mutation background is BRAF. I think approximately 70% of all melanoma cells have BRAF mutations. So because of that, all the melanoma-related essential genes always link to BRAF. But when we run for the same genes, the CEN analysis, we see that, for example, for SOX9, MITF, and ZEP2, there's no direct link with BRAF. While BRAF is mutated, BRAF is essential in skin, but because in skin cells, SOX10 is always expressed, SOX10 is essential for skin cells. Because of that, SOX10 is not related with BRAF because of BRAF mutation. SOX10 is always essential for the skin cells. So we can differentiate this kind of relationship between the essentialities. So we can show them with the CEN. So what about the second part? In here, what we did is, because BRAF mutated melanomas generally very well studied, NRAS mutated ones are like the 30% of the melanomas, and they are not that well studied. So what we did, we take the NRAS mutation related essential genes and we check their actual protein-protein interactions, if they are interactive. Because our assumption was, if they're essential when the NRAS is mutated, there should be some pathways enriched in there. There should be linked together. So we saw that, yes, they are linked, and they are mostly enriched in RAS signaling pathway and also mTOR pathway. And also we know that while NRAS is mutated, RAF1 and SHOC2 from the previous studies, we see that signaling go through RAF1 and SHOC2 while NRAS is mutated. So we solve them in the pathway analysis too. So also another interesting point is IGFR1. This is receptor tyrosine kinase. So it is also enriched. It is also essential when there is NRAS mutated, but when the BRAF is mutated, like the other melanoma cell lines, it's not essential because BRAF is always active and it doesn't need any upstream signaling. So it really makes sense. These are the examples that how we can actually use CENs to identify different biological to answer, or use CENs to answer the biological questions. So for a discussion, CEN-tools identify core genes and context-dependent essential genes, and using our website, you can able to navigate through different specific dependencies, or you can create your own CENs for specific tissue and cancer type. Or if you have any other studies and, for example, signaling-based studies, you can directly use the essentiality association to integrate with it, because as we showed that there are meaningful results coming from. And also, if you have your own data, you can use our Python package. I would like to mention a little bit about it, which is PyCen, and it's in our GitLab repository. So PyCen is give the opportunity to users to search their own data to find association. So how you can use this package? You can simply download it from the GitLab and install the requirements, and then you can use it like Python tree, PyCen, and the input. And these inputs are really detailedly written in our README file. But I will give a little bit examples. For example, the Python tree, PyCen, and then I'm just saying that, okay, for BRAF gene, use the mutation context for the cogene BRAF, which means that don't look all the genes for BRAF mutation, but look only for BRAF to BRAF association to see if there is any or not. Group is tissue, and project is brought in hotspot mutation use only, and write the CEN file into there. You can also give your own essentiality data or own expression data, or you can simply create gene to cell line specific data. For example, okay, there is MSI positive, MSI negative. You can look at it as we did in our third examples. And you can give your own cell lines if they are in our essentiality table, or simply you can create your own essentiality table to put it here. So this is only for the users if they want to run their own analysis. So this is all from me, but I would like to thank my amazing mentors, Evangelia Petralaki and Sumana Sharma, and they were like amazing. And I really would like to thank Paula and her contribution and all of her effort for creating our website. And also I would like to thank Gavin Wright from Wellcome Sanger Institute, because I didn't show, but he opened his lab to validate some of our results in the wet lab. And I would like to thank all the Petralaki Research Group for their support and feedbacks and everything. And for our funders, Emble and Wellcome Trust, you can find our paper with this. And also, this is our website that you can search through the essentiality and associations. And this is our GitLab repository. You can find PyCen. This is under the Petralaki lab page. If you have any questions, please feel free to write in the chat box. I will just look at them now. And thank you for listening, and thank you for your time. I'm ending the show right now. Okay. Thank you, Zeynep. You write from private, I think. For the presentation, actually, I can send them, but all of them are in our paper, so you can find them. But if you want, I can send my slides too. I can send RSG group, and maybe I don't know how we can do this. Hello? I don't know if I'm seeing. Okay, I now see the questions. Muntader, hello. Well, listen. Intronic, yes, it could be, but we only take the missense mutations. But for the Muntader asked the intronic mutations and their effects. We didn't use, because we only use the missense mutations. for our mutation analysis, but as I said, you can create your own discrete mutation data, and you can use the Python to create the intronic mutation essentiality across genes. So yeah, but we didn't do it. Thank you, by the way. Sorry. Hi, Amy.
Hi. Can you hear me? I guess I'm having trouble with my microphone right now.
Yeah, I can hear you right now.
Okay, so no problem. Now we have another question in Q&A session.
For trained data, actually, we only identify, separate them. But for the remaining part, it doesn't link exactly with the trained data because it's only the association of the genes. But essentiality data is from cancer cell line, human cancer cell line. So if you want to use this data for different species, most probably you need to give the essentiality values of that species, because otherwise it doesn't make much sense, I think. But for example, if you have all of your data, then why not? Yes, you can, because it's just it only look at the structure of the data and what is the proper test, and apply the test and link the significant associated together to create a network file.
Yes, if you are currently writing a question, if you can just click on the hand icon button on the right-hand side of your control panel, so we would know that there is a question. Okay, we have another question right now.
Hello, Evrim. Because I think we use the non-parametric tests, yes, I agree with our prediction is not that strong as the parametric test, but at least we're sure that we are not using the wrong test. So I don't think if we use the non-parametric test on normally distributed test has a really bad effect, but I'm not a statistician, so I'm really not sure. But I always think that using non-parametric tests is an advantageous using the parametric version. But yeah, we can, but if we use like tested, and if they are normally distributed and all the variance things is okay for using the parametric test, and if we use some of them parametric but some of them non-parametric, this is affect the network representation because we are combine everything. So actually I'm not preferring using different tests. I don't know if there are any other opinion. Please write the opinions too, not only the questions. It would be great. Oh, you're deleting. I was like, what is?
No, I'm moving them to answered part.
Oh, you didn't. You're not seeing the questions. I'm really sorry. Evrim asked. I'm not reading it because there's no question, but she simply asked if the data is normally distributed and you are using non-parametric test, if it affects the results. And the other question, if they can be used for different species, the scripts, but the data is for human-based values. So I hope they are. Thank you, Elena. And yeah, it is, I think, under the, nobody can see the chat and questions, or okay.
So if you are writing your questions, please use the chat box so that everyone can easily see them.
And there also now there are some missing part in this CEN-tools, and like confounding effects, and scripts can be make it like it can be improved. So they are preparing, I think, CEN-tools version 2. Now I'm not at the group. unfortunately, but they are working on it. And also for the protein-protein interaction part is just an example. And Paula actually for her PhD studies working on it, and she found really good results. And I'm pretty sure that they will like publish it.
I guess we have no questions left.
Yeah, I think so.
Okay, then thank you so much, Cansu, for this very nice talk, and it was very inspiring. And I would like to thank our attendees as well for the participation. And I would like to remind all of the attendees that you can follow us in Instagram. I'm sorry, not in Instagram, in LinkedIn, Twitter, and Facebook. Also, if you register to our BioInfoNet project, then you can receive our invitations for the next events. So thank you so much. I would like all a good evening.
Thank you, everyone.
Thank you for all for your contributions.